Dear students, in previous class I have discussed Mendel's monohybrid cause, law of segregation, etc. Today I discuss the dihybrid cause and the related matters of monohybrid cause. You see, Mendel experimented with the 7 characters with their contrasting traits, and from his monohybrid causes he opined in a principle which is termed as law of dominance. Law of dominance. It states in a hybrid, in presence of 2 contrasting traits, only the dominant trait is expressed in phenotype and the recessive trait does not express in phenotype but remains apparently lost condition. You have already learned it. That is why in all the monohybrid causes Mendel obtained only the dominant trait in F1, and here you see that you practiced already, that is tall and dwarf. Mendel obtained only tall plants in F1. Now, from the knowledge of your monohybrid causes, you may given some problems to solve. Suppose you are given a hybrid tall plant is crossed with a pure dwarf plant, then state the result in next generation. In such cases, you start from where it is given. That is here given the hybrid tall plant. So you start with genotype of hybrid tall plant. Genotype of pure dwarf pea plant. Now you may question, how do I observe which trait is dominant, which trait is recessive? It is a good question. You know the terms hybrid, pure, these are the genotypes. Here it is said that is hybrid tall. That means the tall trait is expressed in hybrid condition. So it is the dominant. Here there is no problem. In other cases, if a capital letter is given after the trait in bracket, that means this trait is dominant. Otherwise, it is said that is it, this trait is completely dominant over the other. Now come to this point. Capital T, small t, the male, small t, female, the parental generation. You know here the 2 types of gametes are formed, capital T type, small t type, and here only 1 type of gamete, small t, gamete. And then if capital T fertilizes the capital T, then capital T, small t. If small t fertilizes the small t, then small t, F1. It is the next generation. No further cause can be done. So result is phenotypic ratio tall is to dwarf equal to 1:1. Similarly, genotypic ratio hybrid tall is to pure dwarf equal to 1:1. Okay, these are the varieties problem you may given, but the solution system the same. Now the 2 other terms I discussed, the back cross and test cross. Back cross and test cross, back cross. When F1 individual is crossed with any of the parent, it is called back cross. For example, in Mendel's monohybrid cross, that is hybrid tall plant in a of F1, that it is crossed with any of the parents, either capital T or small t. So it is crossed with capital T or crossed with small t. It is the back cross, and this cross you must practice or done in your class tasks. And test cross, to know the genotype of the unknown individual with dominant trait. Then it is crossed with the homozygous recessive parent. So to know the unknown genotype of an organism with dominant trait, then it is crossed with homozygous recessive of the parent. Suppose in Mendel's experiment you are given a tall plant. The tall plant, we know the tall is the dominant trait, and the given plant may be either pure or hybrid. So from this test course we come to know what type of plant is given to me. It is then crossed with the homozygous recessive, because you know the homozygous recessive only produces one type of gamete, the recessive gene. Suppose you are given the pure tall plant, then it is crossed with homozygous recessive. The tall plant produces the capital T gamete, small t, and dwarf plant small t gamete, and due to cross in F1 all the plants obtain the tall. So we can say the given plant is pure tall because there only the capital T genes are present, and hence in presence of capital T gene the recessive gene cannot express. And if it is the heterozygous tall, then the cross is done. Here two types of gametes are produced, capital T type, small t type. If capital T fertilizes the small t, then capital T small t tall, and small t then dwarf. So here dwarf plant is obtained. That means the dwarf gene must be present in the given plant. So it is the heterozygous tall. Okay, now we come to the point, the dihybrid cause. The Mendel's monohybrid cause, Mendel took only one character with its contrasting trait. In dihybrid cause, he took two characters with their contrasting traits. So when a cross is done between two organisms of the same species, considering two characters with their contrasting traits, it is called dihybrid cause. So Mendel's in experiment, the selected plant, sweet pea plant, selected character. There are two characters. Number one, the shape of seed, contrasting traits. Around capital R and wrinkled small r. So you see the round, it is the dominant, wrinkled small r, it is the recessive. Okay, like shape of seed and color of cotyledon. color of cotyledon, not color of seed. Cotyledon of seed, color of cotyledon, contrasting traits, yellow, capital Y, and green, small y. So yellow is the dominant and green is the recessive. So Mendel took the two pea plants. One pea plant produced the round and yellow type of seed, and another plant produced the wrinkled and green type of seed. And so the seeds that are produced, here we write only mentioning the seed. So genotype of pure, the parental generation starts with pure, the pure round and yellow, pure round yellow, the round and yellow actually nothing but the monohybrid cause. Suppose in monohybrid cause you would write the genotype of pure round, then or genotype of pure yellow, and as the two characters we take, these characters were present at different homologous chromosome pair, and that is why we write in separately. It is one pair of chromosome, it is another pair of chromosome. Similarly, genotype of pure wrinkled green seed, small r, small y. Okay, now cross. So like Mendel cross between pure round yellow and pure wrinkled green seed containing pea plants. So round yellow male, wrinkled green female, symbol P, gamete, meiosis and segregation. During gamete formation, from each gamete receives one chromosome from each pair. So here two pairs of chromosomes are present. A gamete receives one chromosome from each, that is from capital R, from capital Y, Y. Since these are the similar type of chromosomes, so only the capital R, capital Y type of gametes are formed. All the gametes form the capital R, capital Y type. Similarly, small r, small y. In reality, gametes are not 1, 2 or such. You know, during meiosis, the one cell divides to form 4 haploid cells. Here also from this cell, the 4 haploid cells are formed. Each gamete bear the R and Y type of. Here we write only the type, RY type, small r, small y type, not one. Now cross fertilization. So capital R, small r, capital Y, small y, F1. Here, the capital R, small r, capital Y, small y, you write R and R together, Y and Y together, never R, Y in such a way, small r in such a way, because these are the homologous chromosomes. When R and R come in the zygote, they form pairing. So write in such a F1. In F1 he obtained the plant and the seeds, all around yellow. Then he allowed the self-pollination among the F1 progenies, like capital R, small r, capital Y, small y, capital R, small r, capital Y, small y, male, female. These are not the plants. These are the male and female sex organ of a flower of all plants. In all plants, it is the bisexual flowers are present, and these bisexual flowers contain both male sex organ, female sex organ. It is the male sex organ, it is the female sex organ, and that is why self-pollination. Okay? Now gamete formation. You already learned that is from each pair the gamete receive one. So here the one pair, two types of chromosome, that is R and small r. So capital R will receive any of these, that is either capital Y or small y. So capital R, capital Y. Similarly, capital R, small y, small r, capital Y, small r, small y. Here also the similar four types of gametes. Capital R, capital Y, capital R, small y, small r, small y, like gamete. Now you follow the gamete formation. Because during gamete formation, there is a formula, how many types of gamete will be formed. It is the 2 to the power n. N is the number of contrasting pairs. In monohybrid cause, the number of contrasting pair 1. So 2 to the power 1, two types of gametes are produced. That is where you get the capital T, small t, two types of gametes. Here the contrasting pairs 2, capital R, small r, capital Y, small y. So 2 to the power 2, four types of gamete will be produced. And during gamete formation, you follow this rule. Do not do so. It is a bad habit in this way. And this type of cross mainly done during the experiment. You know from capital R, small r, capital Y, small y, from each pair, one chromosome enters one gamete. You take the first letter. Always remember, in all gametes, the first two letters, out of first two letters, one letter must be present. So you take the first letter. Then from the second pair, you take any one of it, and then the next one. Generally, at first you take the capital letter. So the capital R, from another pair, capital Y, and then capital R, another pair, small y. With capital R, thus with the two chromosomes of the second pair combination completed. Now you take the next one. Now the small r, similar way, capital Y, small r, small y. If you are given the three pairs of chromosomes, suppose capital A, capital B, and capital C, small c. So here 8 type of gametes produced, 2 to the power 3, 2 into 2 into 2. You follow the same rule. You take the capital A, and then from the second pair the capital B, and from third pair capital C, then capital A, capital B, and small c. So with capital A and capital B, all types of combinations completed with the capital C. But with capital A, small b combination is not formed. And now you write the capital A, small b, capital C. Capital A, small b, small c. With capital A, all the combinations completed. Now you follow the small a. Similarly, small a, capital B, capital C, small a, capital B, small c, small a, now small b, capital C, small a, small b, small c. Suppose you are given in such way, that is capital A, small a, capital B, and then capital C, capital C. Well, how many types of gametes should be formed? The 4. Because here number of contrasting pair 2 to the power 2. Here you follow the same rule. Capital A, capital B, and place the C. Capital A, small b, then small a, capital B, small a, small b, capital C. Completed. Okay? Now come to the cross. 4 types of gametes are formed. So 4 into 4, 16 combination will be produced. So you draw the checkerboard. It is the placement for gamete, male, female, 1, 2, 3, so 4, 1, 2, 3. Now you place the gametes, capital R, capital Y, capital R, small r, capital Y, small r, small y. Similarly, capital R, capital Y, capital R, small r, capital Y, small r, small y. Now you place the finger in this and then cross with these gametes. Like capital R, capital Y, capital Y, capital R, capital Y, small y, capital R, small r, capital Y, capital Y, capital R, small r, capital Y, small y. Now the second, the capital R, capital Y, small y, capital R, small y, capital R, small r, capital Y, small y, capital R, small y, small y, capital R, small r, capital Y, capital R, small r, capital Y, small y, small r, capital Y, small y, small r, capital Y, small y, capital R, small r, capital Y, small y, capital R, small y, small y, small r, capital Y, small y, small r, F2. From this cross, now we find out the result, the phenotypic ratio and genotypic ratio. You find where you see the 1 or 2 capital R, it is round. And where you see 1 or 2 capital Y, it is yellow. In case of wrinkled, it is the 2 small r. In case of green, it is 2 small y. In this way, the round yellow, it is the round green, round yellow, it is the round green, round yellow, it is the wrinkled yellow, round yellow, round green, wrinkled yellow, wrinkled green. According to the Mendel obtained the phenotypic ratio, a round yellow is to a round green is to a wrinkled yellow is to a wrinkled green, 9:3:3:1. Now the genotypic ratio. From genotypic ratio, to find out the genotypic ratio, you follow this. That is, first you write the capital R, capital Y, capital Y. In this checkerboard, the capital R, capital Y, present only 1. So 1 is 2. The next one, capital R, capital Y, small y. In this checkerboard, it is present, that is this genotype present 2. Then capital R, small r, capital Y, also 2 is to capital R, small r, 4, 1, 2, 3, 4. These are the round yellow, and in phenotypic ratio you say the round yellow 9. 1 plus 2, 3 plus 2, 5 plus 4, 9 is to now capital R, small y. It is the round green. The round green 1 is to capital R, small y, 2. You follow the round means either 2 capital R or 1 capital R, small r. In this checkerboard, where the round and green or wrinkled yellow, where 2 capital letters are present, then it is 1. And when 1 capital letter, 1 small letter, that is when pure dominant and pure recessive, it is 1. And when hybrid dominant and pure recessive, it is 2. So 1 is to 2. The similar way, small r, capital Y, 1 is to small r, capital Y, 2 is to small r, small y, 1. So 1 is to 2 is to 2 is to 4 is to 1 is to 2 is to 1 is to 2 is to 1. And from this he observed that in the dihybrid cause in reality nothing but the monohybrid causes which followed the ratio of monohybrid cause also, and it is the explanation of dihybrid cause. It is discussed later. Now you say come to the point. From dihybrid causes, Mendel formulated his second law of independent assortment. From this law, he stated that in hybrid condition or hybridization, when 2 or more pairs of factors, that is gene, assemble in a hybrid, then during gametogenesis, each factor of a pair combines with either factor of another pair independently and in all possible combinations. It is the independent assortment. That is, in this experiment, in a hybrid, 2 pairs of factors assemble. And during gametogenesis, when they form gamete, each factor of a pair combine with either factor of another pair independently. That is, capital R either with capital Y or capital R with small y. Similarly, small r with capital Y or small r with small y. And one factor cannot influence the other. That is, one pair never influence the other. That is, in monohybrid cause, you see the tall and dwarf, they express separately. But in case of dihybrid cause, 2 pairs of characters are present, and one pair is the round, another pair of chromosome where the yellow present. Round cannot influence the yellow, or yellow cannot influence the round. Okay? And now explanation. The Mendel observed that is the one character cannot influence the other, and from dihybrid causes he obtained the following results. like a round yellow, round yellow, 315 seeds. A wrinkled green, 32. Both are parental types. That is, we start the cross with round yellow and wrinkled green seed. Mendel, and also the round green 108 and wrinkled yellow 101. From this result, these are the recombinant type. Recombinant type, that is newly formed. When Mendel started the cross, only these two types of plants he took, and due to such cross he obtained the new two types, round green and wrinkled yellow, recombinant type. And from this result, he not only obtained the phenotypic ratio, the 9:3:3:1, but also he observed that these followed actually the monohybrid ratio. For example, suppose the round 350 and the round, here also round, the plus 108 is to wrinkled 32 plus wrinkled 101, and there is 3:1. Similarly, the yellow 315 plus yellow 101 is to green 32 is to plus 108, 3:1. And reason behind this, the Mendel said that during gametogenesis, capital R of a pair, that is one factor of a pair, only combine with either capital Y or small y, never with small r. So two types of gamete. Similarly, small r combine either with capital Y or small y. So two types of gamete, total 4 types of gamete, and 4 into 4 due to random fertilization, that such combinations are formed. And from this experiment he obtained that the dihybrid cause nothing but the 2 monohybrid causes, and here he also find that one character never influence the other characters. Also these factors, that is character, must be present in 2 different homologous chromosome pairs. Now from these dihybrid causes you must do as homework from the animal, that is guinea pig. In monohybrid cause you took the character, that is color of hair, contrasting trait black and white. Here you take the 2 character, one is the color, contrasting trait black and white, black dominant, and another is the nature or shape of hair, rough, capital R dominant, and smooth, smaller. So the genotype of pure black rough and genotype of pure white smooth And now follow the same rule that you now learn about the dihybrid cause. But after F1, when you take the cross between the two F1, here you write the F1 offsprings, only F1 offsprings. Okay, now the dihybrid cause is completed. From his experiment, both monohybrid and dihybrid causes, Mendel concluded the following. Number one, he said that different characters or traits are controlled by factors, and each factor carries the code of a trait, and these factors remain in pairs, and these pairs may be homozygous or heterozygous. Number two, each trait is controlled by a pair of factors. Number three, each gamete obtained one factor from the pair. And number four, during fertilization, the paired condition is restored in the zygote. Number five, when zygote develops into an offspring, then these factors particularly expressed, that is when dominant is present, it is expressed, and out of two factors in a zygote which is expressed in the offspring, one comes from the father body and the other comes from the mother body. These are the conclusions. Now, next topic, the incomplete dominant, that is deviation of Mendel's experiment. Now I discuss it in the next class. Now, dear students, teachers, respected teachers, guardians, etc., if you like it, then share it or give the comments and like, etc., so that I may inspire from you. Okay, thank you.